Thank you everyone today for joining us for today's webinar. Good morning, good afternoon. Our webinar today will be Complete Your R&D Transformation with Intelligent Sample Traceability. And my name is Rashida Kar, and I'll be your speaker and your moderator today. I'd like to introduce today's other speakers, Ashoka Rajendra and Dima Gharaibeh. A little introduction about me. I'm part of the product marketing team at Benchling. I've held various commercial and research positions in the past at Forte Bio and Genentech for the last 10 years. I've done my bachelor's and my master's in immunology from McGill University. Ashoka Rajendra, he's the head of product for Benchling's registration and inventory applications and works with leading biotech organizations to optimize their R&D processes. He studied biology at UVA and has since worked on software products for genomics and life sciences. Dima Gharaibeh, she comes to us from Sorrento. She's a project manager and a senior associate scientist for the Oncolytic Immunotherapy program at Sorrento Therapeutics. And she serves as the administrator for Sorrento's R&D-wide Benchling deployment. Prior to this, Dima has worked in various settings, including academia, federal, and industry, planning and executing in vitro and screening assays for infectious diseases. She's a strong advocate of data curation as a means to efficiently track, organize, and execute program objectives. Before we begin today, I'd like to go through some housekeeping items. Please feel free to submit questions at any time by clicking on the Q&A button at the bottom of your screen. We will reserve time at the end to answer them. If we cannot get through the questions today, we'll be sure to follow up via email. Additionally, we'll be polling the audience today through the content of today's webinar through multiple choice questions. If you could select the response that applies most closely to you, that'd be really helpful for us. The first polling question should be popping up on your screen right now. The first question is, are you currently using or have used Benchling in the past? Five more seconds. Thank you so much. So it looks like we have a good mix of about 60% current Benchling users and about 40% who have never used Benchling in the past. So we're excited to kind of have both of you here today. So at Benchling, we feel we're enabling the next phase of digital transformation of life sciences R&D. Our talk today will be divided into three main sections. The first section will consist of really discussing the data management challenge in life sciences R&D and how the pace of innovation has been astounding in the last decade that's led to a number of new challenges. The second section will cover Benchling's unified sample traceability solution and how that will help address and solve for these data management challenges. And our third section will be covered by Dima. We'll have a case study and also demonstrate how Sorrento Therapeutics' success story with Benchling. So before we head over to really discussing and deep diving into the data management challenge in life sciences R&D, we'd like to poll the audience for the second question of the day. The second question is, what is a primary way you're currently recording research data across your organization? Thank you. It looks again like we have a pretty good mixture of ways of recording data, including ELN, LIMS, Excel. So we're going to be discussing that a little bit more in detail now. All right, so we're entering a new era in life sciences. As I mentioned before, the pace of innovation and scientific development in just the last decade has been staggering. These last 10 years have come to fruition with landmark occasions, whether it's the invention of CRISPR gene editing, ubiquity of next generation sequencing, to the approvals of CAR T and gene therapy drugs. There's also been significant progress beyond biopharmaceuticals, including cultured meat becoming mainstream and new biomaterials. These advances are changing how we feed, how we fuel, and how we heal the world. Starting with the way how we feed the world, raising livestock is one of the most environmentally expensive processes. We're building towards a lab-grown meat future. Life sciences is also transforming how we fuel the world. We're leveraging the potential of microorganisms such as yeasts and plants to produce fuels and other biomaterials. And most importantly, we're also transforming how we heal the world. Over the last 60 years, the treatment of disease has primarily relied on chemical synthesis drugs that were designed to treat the symptoms rather than the underlying cause. We're now moving towards personalized medicine that's directed at the cause agents of the disease. While these innovations have been great, they're also making R&D increasingly more complex. Companies have historically turned to a large number of point or single purpose solutions mixed with in-house apps, ELN, or LIMS tools, as we saw in the last polling questions, to stitch together your workflows. Current R&D data management tools are really falling short in meeting these new complexities, whether it's difficulty in finding data across systems, or assembling and aggregating data, or really having any kind of standards in entity naming or processes, and most importantly, handoff between these specialized teams. As a result, scientists and R&D leaders are left to accept blind spots in their workflow, or spend more time piecing together data. We often hear from our customers that scientists spend 40 to 50% of their time searching data, aggregating data, and cleaning up data. This has to change so scientists can do what they do best. It's time for a third polling question for the day. The third question is, what is the biggest bottleneck in your workflow, i.e., where do you spend the most time? Five more seconds. This is great. So it looks like majority of people are spending their time collecting and aggregating data for reports or looking for historical data and information. So we're here to tell you how we can help solve for those challenges. So Benchling's mission is really to accelerate life sciences R&D. A company has been founded in 2012 with a team of scientists and engineers who were frustrated with the poor quality of the software they had available to support their research. So with the support of top-tier investors, Benchling built out a range of applications built on a common unified platform that closely followed the needs of our customers. These 7 applications include the molecular biology, the notebook, the registry, the inventory, workflows, requests, and our newly launched insights. The big main thing here is all of these applications share the same configurable data model and can be accessed from a single interface. That really makes it easy for you as scientists to leverage what you need without leaving the software. We're fortunate to have been working with over 300 innovative life sciences companies, including many of the world's largest enterprises. These companies are leading the digital transformation with Benchling, and our team is committed and well-equipped to provide the best possible solution to support you. So diving a little bit deeper into Benchling, we believe that the first phase of R&D transformation was the adoption of ELNs from paper notebooks, or utilizing multiple disconnected specialized applications that did exactly what you needed it to do. And this was successful. It simplified note-taking, it improved data searching and sharing, and also helped recording potential intellectual property. However, these solutions are not sufficient on their own. They are only a component of a larger informatics puzzle that's needed to truly help address the challenges and reshape R&D. Which is why we believe that the next phase of R&D transformation requires a modern and a unified solution that's flexible to adapt to your evolving needs and is also able to crack and interlink the entire R&D process, from molecular design, experimental tracking, data acquisition, as well as process management. We believe that for this unified platform to be successful, it's important to build the right foundational elements for capturing, centralizing, and integrating all types of structured and unstructured data. In Benchling, the three apps that constitute as the core of our platform and really enables these foundational elements to be set are the registry, inventory, and the insights. And these are what we're going to be focusing on in today's webinar. The Benchling Registry, Inventory, and Insights app enable modeling and tracking of all your complex molecules, their underlying components, their physical locations, and tie all that to the experimental context and results. This centralized information is then able to be queried to draw insights across experiments and programs to really drive your R&D forward. So let's deep dive into these three applications. The first is our registry. The registry is really able to model anything with end-to-end traceability. What we mean by that is that it has flexibility in being able to model not just simple molecules such as mAbs, but also complex molecules such as ADCs, bispecifics, viruses, and also, as we know that the complexity of these modalities are increasing, it's able to adapt as your organization grows. And today, in Dima's section of the talk, you'll see how they've adapted the registry to not only just register viruses and mAbs, but also presentations and legacy data. The registry enables mapping of relationships and truly tracing the lineage. So you have complete visibility from sequence, plasmid, the cell line used, the final product, as well as the in-process entities. It's also able to capture structured data and hoist that against all of these different entities for in-process as well as final molecules. The inventory serves as a digital window into your physical lab. It's able to interlink sample information, such as if you have a physical lot that you produced and you've put that in a fridge or a freezer, it's able to link these physical lots to all that experimental context and how it was produced, as well as all the results that were gathered or captured against it. It's able to integrate with barcode scanners and smart fridges and freezers to constantly keep your samples up to date, so you never will lose a sample again. And the inventory really streamlines communication, especially when you need to submit assay requests. We see now with R&D becoming more complex, there's a lot of specialized groups, and often the assay group is different. So if you need to submit those samples, you're able to submit these assay requests directly from the inventory applications, so they have all the information of where the sample is located and all the information around it, related information. And the insights application is truly what we call the intelligence that will help power your R&D. It'll help you generate scientific insights because it's able to aggregate and collate information so you can build reports to understand and compare results within or across experiments. It's able to unlock operational insights because you can dive deeper into resource allocations and productivity and truly optimize your resourcing. And also drive collaboration because you can make these individual queries or dashboards and share that within your team or with leadership to make R&D data-driven decisions. So what we mean by this is now you need a really important foundational elements to truly solve for this big informatics puzzle. And what these values are unlocked from having a solid foundation is you have data centralization and standardization across each step of the process, truly creating a single source of truth that's searchable and queryable. It increases scientist productivity because it reduces non-value added tasks, such as transcribing data into multiple locations, manipulating Excel files before uploading into a LIMS system, or really sending multiple emails when you're trying to do some kind of handoff or requests. It also helps improve data integrity because you've now standardized entity naming and processes that reduces redundancies or mistakes that could be made by changing the name slightly so you weren't able to aggregate that data before. It minimizes this chance of human error, truly promoting data integrity. And data centralization on Benchling will really help unlock the ability to glean insights across experiments and programs to make these data-driven decisions. So now we want to dig deeper into a scientific use case, which is going to be protein production. Ashoka is going to be walking you through the Benchling platform to demonstrate how we can show intelligent sample traceability with Benchling.
Thank you, Rashi. Go ahead and share my screen. All right. So as Rashi mentioned, I'm going to walk through the product on a scientific use case, protein production. So what I have here is a reasonably simplified protein production workflow where my goal as a scientist is to design media for an optimal yield. I want to optimize for cell growth and protein yield. So first, after determining a candidate molecule that I want to produce, such as an antibody or some other protein, I'm then going to design media to optimize for cell growth. I want to prepare that media in the lab and record the process of preparing that media to ensure it follows the correct QC processes. I'm going to use that media to grow cells in a bioreactor and combine that with plasmids that express the proteins for the molecule that I seek to produce. As these cells are growing in a bioreactor, I'm going to record a cell count and protein yield. And at the end of this, I'm going to analyze this data to select optimal growth conditions to determine if the media I'm using or that I've designed is optimal for cell growth and protein yield. As Washi mentioned, a registry, an inventory, and insight system is the foundation for a digital transformation in the lab. And going through this scientific process, I need to keep track of many different kinds of samples. When I am selecting my candidate molecule, which could be an antibody, this is comprised of chains expressed by plasmids. When designing media for optimal yield, I need to design a media recipe and have cell lines associated with that. When I am actually preparing the media in the lab, I need to record the actual quantities of the ingredients that I prepared, as well as QC results such as pH and osmolarity as I prepare this media. When I'm growing cells in a bioreactor, it's really critical to know which cell line lot I used, what prep of media did I use, and what plasmid preps were transfected into the cells. And at the end, I need to record experimental results such as cell count, protein titer, and antigen binding affinity. At the end, the point of recording all of this information is to narrow in on the optimal growth conditions. I'm actually going to jump into the Benchling product now so you can see this in action. So for those of you who are unfamiliar with Benchling, Benchling is a web application. What you're looking at is a notebook entry in Benchling. Along the side are various applications. we have our insights tool, process management tools like requests and workflows, our inventory system, our registry, and so on and so forth. And today we're going to be focusing on our registry, our inventory, and our insights tool. What we're going to do is go through each process of this experiment step by step. And in doing so, illustrate some of the features around sample traceability as well as insights. The first step is to determine our candidate molecule. In this case, this is an antibody. This monoclonal antibody is comprised of two different chains, a heavy and a light chain. And in Benchling, I can define a schema or a structured data format to determine what kinds of data I should record about this new antibody. I can drill into this antibody to look at the heavy chain that comprises it, the CDR annotations to overlay onto the amino acid sequence, as well as the biochemical properties of this amino acid sequence. I can also record structured data, such as what kind of an antibody it is. And benchling using sample intelligence will also include things such as related items, such as the plasmid that I can use to actually produce this antibody in the first place. So here I've drilled into the plasmid with its DNA sequence, the plasmid map, as well as additional structured data fields, such as the chain that it actually expresses, bringing me back to the protein that I intend to create. The goal of this experiment is to design media for an optimal yield. And you could represent this as a table in our notebook entry, where I might outline a media recipe with DMM high glucose, FBS, and L-alanine and glutamine, associate catalog numbers so I know which vendor and manufacturer it came from, and specify the amount in units to comprise this recipe. You can also record this information as structured data in Benchling. So as you'll see in the center here, each of these ingredients is represented as an entity in Benchling, associated with its catalog number, target amount, and units. I can also include additional structured data, such as the form that it should be stored in, storage conditions, as well as QC metrics such as pH and osmolarity. Now that I've designed a media recipe, I need to actually prepare that in the lab. Recording the preparation process of media is critical, and recording not just the conceptual versions of things, such as your antibody molecule or your media recipe, but also the actual physical samples you create in your lab is important to ensure that your experimental conditions meet QC boundaries. So my objective here is to prepare an iteration of this media. So I'm going to modify levels of L-glutamine and add a mystery ingredient that I hope will improve the yield of my cells. So what I'm going to do is actually create a prep of this recipe. And what you'll notice is I've inherited all of the ingredients from my recipe. So I have the ingredient, the catalog number, and then a computed target amount based on the fact that I want to make 2 liters of this recipe. But what I can do is also enter in measured amounts in the lab and include source. So you could imagine this as being your lot number. But because I'm running an experiment, I want to add in a mystery ingredient that was internally prepared. And I'm going to add 5 grams. So I can record the structured information, not only creating media based off of recipes, but also iterating on media based using a recipe as a template. Back in my notebook, after I prepare this media, I can also record QC metrics, such as pH and osmolarity, so that when I come back to this media in the future, I can view the experimental results that were recorded and involved in its preparation. Now I'm actually going to grow cells in the bioreactor. To do this, I want to specify the experimental conditions for this bioreactor run. And as I mentioned before, my goal here is to keep everything the same except change which media is being used. So I'm going to keep the same transfection protocol, same cell line passage, same plasmid lots being used in each one, and I'm making the same molecule. But the media prep being used in each bioreactor run is different, with the first one, as we just stepped through, being one with a mystery ingredient added to see if this improves cell growth and protein yield. Once I actually run this experiment, I can record experimental results directly in the notebook or with instrument integrations. So here, I've chosen to upload a spreadsheet of experimental results, recording time series data for each of my bioreactor runs. So I'm recording for each bioreactor run metrics like pH and glucose, as well as the cell density, and recording this directly against the bioreactor run that I've just created. After I've done my bioreactor run, I want to harvest these samples and store them in inventory. So for bioreactor run, that's producing the same candidate molecule, I've produced a new antibody lot. And this represents the actual physical sample in the lab. This antibody lot will go into storage. So I can actually open up this box right here and show a filled out box with, a couple dozen empty positions. And what I can see are all the tubes and volumes associated with these antibodies. So I've just created a set of new antibodies, decided to put them in storage. And for each of these things, I can click in, see the concentration, when it was made, and click into the entity itself and see everything about this antibody lot. So as Rashi mentioned, this is really about sample traceability. From the molecule I created, I can see where it's stored, in which box and which freezer, but I can also see all of the information about how it was created. So I have the media prep that was used to create it, the plasmid lots, the cell line passage, as well as the chains that actually comprise this protein, all hoisted onto this antibody lot. So you have full end-to-end traceability. I can also refer to which notebook entry this antibody lot was created. Lastly, after I've harvested the proteins from this, what I really wanted to determine was whether or not my new mystery ingredient actually helped with cell growth. So what I can do, and the point of recording all of this structured information, is to analyze this information using insights. So I can create queries, such as the one you're seeing, defining them using SQL, or just having out-of-the-box parameters, and look at each media that I prepared, media 1 being the one with the mystery ingredient added, to determine how effective it was at growing cells and producing proteins. It looks like the mystery ingredient wasn't particularly effective and actually led to very poor cell growth, whereas the other media preparations, which did not have a mystery ingredient added, seem to work fairly consistently with one another. So with this brief demo, I hope you are able to see how Benchling is able to flexibly model the variety of different samples that you're working with in your lab, represent the complex relationships to give you full sample traceability, and also allow you to aggregate information to derive experimental insights, all from the same software platform. Before transitioning to Dima, we have one more polling question. The question is, do you feel like you have a solid foundation for capturing and connecting sample lineage, results, and experimental information? unfortunately can't see the poll results, but I'm going to transition this back over to Dima, who can do a much deeper dive into how Sorrento used Benchling to digitally transform their lab and give you a much more scientifically interesting example than the one I just presented.
All right, thank you very much. I'm just going to take a quick moment to share my screen here. All right, thank you for attending. And today I'm here to talk to you about the Benchling implementation at Sorrento Therapeutics. Sorrento Therapeutics is a medium-sized clinical stage biotech located in sunny San Diego, California, and our main focus is antibody development. Our mission is to develop therapeutics to extend and also enhance the lives of cancer patients by reducing the malignancy of life-threatening tumors, improving safety of current treatments, and also discovering novel solutions to make living with cancer more tolerable. Some of our investigational goals include the advancement of monoclonal antibodies against SARS-CoV-2 for COVID-19 treatment and utilization of antibody libraries for oncogenic targets. The Serrano Therapeutics GMAB platform includes therapeutic, bispecific, and monoclonal antibodies that are utilized and developed within our various teams. The platform uses RNA transcription to amplify antibody variable domains from over 600 donors. This library contains over 10 quadrillion distinct antibody sequences and is one of the largest fully human antibody libraries in the biotech industry. Serrano has successfully identified fully human antibodies against over 100 clinically relevant oncogenic targets, including PD-1, PD-L1, and CD38. In addition to the GMAB platform, Sorrento also has some pain management assets, which includes RTX, and this is derived from a particularly spicy plant to produce a non-opioid single injection treatment for control of terminal cancer pain. A second asset is ZT-Lido, which is a next generation lidocaine patch for pain relief associated with post-herpetic neuralgia, which often affects the elderly, individuals that have suffered a shingles infection, and HIV or cancer patients. So to now specifically focus on the Sorrento R&D groups, which are the groups that are utilizing the Benchling platform. We have 3 antibody-based groups, which are shown here on the left-hand side, and they encompass research operations, technology development, and antibody discovery. Our nanoformulations group develops non-viral gene delivery platforms, and chemical immunotherapy focuses on cell surface engineering and targeted drug delivery. The translational immuno-oncology group tests antibody candidates that are of the highest interest, and our CAR-T groups explore a variety of T-cell and natural killer cell applications. Finally, there's oncolytic immunotherapy, which is the group I work in, and we utilize an HSV-1 modified oncolytic virus against cancer cells while concurrently stimulating patient immune response. Overall, Sorrento has experienced some rapid growth. 5 years ago, we had 48 employees, and we currently have around 300 employees, with 95 of these in the R&D area. As you've seen from our background, we have a pretty unique company with a great variety of research. Within R&D, there are 11 R&D groups, and they're made up of 4 to 18 employees. There can be a siloed workflow due to the unique projects that everyone is involved with. Previous to Benchling, the R&D groups used paper lab notebooks or PLNs, and very few people had experience with ELNs. Groups would complete their PLN entries in a variety of ways. Some of the more tech-savvy would paste in Word or Excel inserts into the notebooks and reference electronic data. while others would stick to hand scribing the entries and had collections of paper binders. And no matter what, a lot of glue sticks were used. So based on this PLN background, we needed an organized strategy to implement the Benchling ELN to this large, diverse group. So with guidance from the Benchling customer success team, we first worked with each group to establish project structure and permission settings. This involved deploying a questionnaire to each group to determine their organizational structure. We then introduced each group to the overall Benchling scaffolding with data organization examples customized to their group needs. Concurrent with this, the ability to set permission ranges and provide ownership and sharing options for each group was extremely helpful. We next had an R&D-wide teaching seminar that introduced the ELN's general use and navigation. And to set up for this, Benchling Access was set up to allow users to navigate with in-time tutorials. After that, there were smaller group seminars organized to present the Benchling Molecular Biology Suite for the scientists utilizing sequencing data. For this, there were select sequencing tools of highest interest featured and presented. And after this, there were PDFs of training notes and tips that were available for both current and new users. Finally, we held planning meetings for the registry structure and implementation. And for this, again, we deployed surveys to establish custom registry needs and permissions for each group. And then the groups met with Benchling to build and refine their databases. One notable registry development occurred with our antibody groups. A registry was essential here because there was high potential for miscommunication of sequences with the current method that utilized everything from Excel sheets to paper forms. The registry setup here was a big task because it involved a lot of people, unique registry fields, and very different and stringent permission settings. And additionally, not everyone was excited about this registry implementation. However, once things were underway and a demonstration was shown of how the antibody data could be organized within Benchling, there was actually a great meeting aha moment where everyone actually became really excited about the process once they could see how things could be organized. The result is now all antibodies are codified, registered, and linked to the relevant information that can be easily shared across the group. One of the scientists also commented that the registry provides excellent confirmation of material, which is essential for scale-up. The registry will additionally calculate results and provides a link to all other relevant information. Another fundamental shift that occurred was in the improvement of compliance and transparency within the Benchling ELN as compared to paper lab notebooks. Corrections and updates in Benchling can be done easily and can be instantly corrected. In a PLN, every edit will require a physical crossout and explanation. Space is vast within Benchling, which is extremely helpful, because in the PLNs, the static pages could often contribute to a loss of detail. Compliance is greatly increased in Benchling. Insights queries can be used to monitor detailed aspects of the entry and review status. With PLNs, this requires actual physical checks of notebooks, which can be difficult and time-consuming. Experiment status is easier in Benchling because this can be easily checked by supervisors, and scientists can both share and co-author different entries. This is more difficult in PLN because first you must locate the scientist and then find their notebook cache, and within that, then find the resultant experiments. Benchling is like a human brain. The registry can provide evolvable organization, relational capability, and instant communication. And this is not the case with PLNs, which again remain very static. The sequencing capability with Benchling has been a great benefit, as this provides central storage, organization, and utility all within Benchling. Previous to this, our scientists were using multiple versions of different online software, and data was stored in our networks, on thumb drives, and in some cases, even paper binders. An interesting and immediate effect of the Benchling implementation was a market evolution in technology through R&D. It became easy to introduce other online databases because the group was already familiar with online functionality, permissions, and @mentions. Super users began to provide really great ideas for different Benchling applications, including weekly update formats, united methods of group communication, and different ideas for the insights queries. Employees that were initially uncomfortable with technology in general began to flourish with Benchling use. Highly detailed experimental reports that supported our regulatory non-clinical submissions became much easier and easily organized with the use of detailed Benchling templates. And very importantly, the ability to create registries specifically for technology transfer of oncolytic immunotherapy legacy materials occurred. This included the ability to code, organize, and store original virus details, establish a scanned archive of paper lab notebooks, and create a unique literature repository for organized access of original work and knowledge. A specific technological benefit that I can personally attest to is the increased project management organizational capabilities. Within Benchling, action items are readily tracked with the use of @mentioned experiments. Weekly updates are submitted through our group, and these can include entries linked to experimental results. Our meeting lineups, agendas, minutes, and presentations are all stored in Benchling in a central organized location that our group can access. Materials costing has become much easier now because I'm able to access and search a huge amount of material within Benchling. And finally, we created an oncolytic results registry. This is in order to easily access details from all our presented data from our oncolytic immunotherapy group. This first initiated with our registry of virus constructs, which is shown here, which codified our viruses and tracks the origin, sequence, and batch information. The virus registry in turn became part of our presentation results registry. Here, presentation results have been organized into the following searchable fields. This can instantly produce the status of queried viruses and directly links to the source and image. So if you've ever had that meeting where a group attempts to unanimously remember if one specific virus was tested in a particular assay and with a certain cell line, we ended up creating a registry just for that. So here, this registry's search filters can sift through a number of parameters, which include items such as the virus, the type of assay, the cell line, or an author. And then the registry lists presentation slides where those parameters occurred. This will also display a preview image of the slide and links to the presentation result directly. We constructed a similar registry to organize and display all of our legacy materials. In this case, the registry operates similar to a PubMed citation search, where there are filters for author, journal, year, or other parameter. And these can be set, and the located registry entity will produce a direct link to the requested PDF legacy material. In addition to the registries, we utilize the capability of Benchling's new Insights tool to track the status of entries and reviews that are produced at the individual, group, and project levels. With this tool, I run weekly queries and can note any patterns of overdue or lingering entries. And this has been a huge help in monitoring usage and output. The implementation of an ELN on such a large scale has been a real chance to learn and grow. Some of the key ways this occurred has been the opportunity to harness the deployment and organizational capabilities of Benchling to foster our R&D growth. For instance, our compliance has increased with establishment of our best use practices for Benchling, a training process, and a mandatory in-Benchling use exam, and additionally, communication of insights queries to team leaders. Our structure and organization has improved with the deployment of team definition and setup and project and folder layouts required for Benchling use. Our standardization has increased due to the use of team protocols and templates that were established. And finally, our chain of custody has improved as well, as registered items can be traced from their conception into process development and all the way into manufacturing. As a Benchling org admin, one other helpful tool that I discovered was to create a decoy user, shown here as our super user, Oscar Valiant. And this has been extremely useful as a tool to view and troubleshoot issues as a regular Benchling user. And finally, one of the more important things I learned was to very quietly try out my new ideas with super users, and they will find the holes. We've now been using Benchling for over a year, and at this time, all our selected R&D are successfully transitioned from PLN use and regularly utilize Benchling. The R&D groups with registries are regularly using and constantly expanding these databases. And we now have a successful program established to quickly train all new employees in Benchling use. Our use queries are run weekly with the Insights feature in order to monitor use and compliance. In December 2019, I conducted a survey in order to gain some feedback from all our Benchling users. And this resulted in overall high, very positive remarks and included mentions of the tech support quality and great ease of use for organization, searchability, navigation, and overall communication. And across the board, people were very happy to no longer have to scrapbook. In January 2020, we ran a use audit using an early version of the Insights tool and conducted a deep data dive of notebook entry and review completion. We were pleased to see compliance with our internal parameters for entry and review submission. And the results also showed a good entry completion effort in response to the pending audit. Looking to the future, we plan to continue fostering and advancing Benchling utility within Sorrento, including virtual office hours and attendance at the Insights webinar. I hope to increase the registry scope to additional groups at Sorrento and advance its utilization within existing groups and databases. We have also initiated registry use for company-wide applications, including our equipment inventory registry. As we run many unique and varied assays, I still plan to initiate further results linking to our existing registry entities. And further implementation of Insights dashboards will be a major driver in the future, with custom queries specific to each team's needs and increased visibility of our antibody parameters. I'd like to extend many thanks to Stefan Siegfried and Dima Gharaibeh of Benchling for their continued support, Dr. Allen, head of our Oncolytic Immunotherapy, for advocating for Benchling implementation, Dr. Alexis Nahama and Dr. Christoph Pedros for their slide and image data, and the Oncolytic Immunotherapy Group and their constant patience as continued Benchling guinea pigs. Thank you all very much.
Thank you, Dima. Thank you, Ashoka. I hope everyone, we were able to successfully demonstrate how the registry, the inventory, and insights can truly serve as the foundational elements for your R&D transformation and really enable you to have that data centralization and standardization so you have that single source of truth and chain of custody. improve your scientists' productivity and improve compliance within your organization, and also give you access to a lot more data-driven insights across experiments as well as across programs. We'll be moving on to our last polling question for the day before we open the floor for questions. And the last one is, are you currently able to easily aggregate data and run analytics to drive decision-making? 5 more seconds. Wow. Okay, so the overwhelming majority of the audience believe that they cannot they're not able to easily aggregate data and run analytics. So hopefully we were able to show you a solution to do that. Again, a quick reminder that you can submit questions by clicking on the Q&A button at the bottom of your screen. And here's your first question. The first question is, the antibody features that Ashoka that you demonstrated, were they new? And what's kind of the roadmap that looks like for that?
Yeah, so I gave a peek at automatic CDR annotation, where you can take the chains that comprise the antibody and generate those annotations. We'll be having a molecular biology webinar at the end of this month, or in the following month, to actually go through these features in more detail. Perfect.
The next question is actually for you as well. It's, what can I use the new mixture features for?
Yeah, so the mixture feature can be used for creating buffers or chemical solutions, media, as well as formulations. And this can both be used in use cases where you seek to strictly adhere to a recipe, where you want to make the same thing every single time. You can also use this to do media development or iteration, such as the one that I showed in the demo today.
Perfect, thank you. Dima, the next question is for you. Could you please elaborate on the additional use cases for the registry that you plan to expand to in the near future?
Yeah, definitely. So not all of our groups are currently using the registry. So with a little more time, we plan to implement that to all the groups, because I think this will be a great benefit for everyone. We all have things we need to organize. I do still plan to begin some results linking with current registries, and this will include some of our more standardized assays. Basic things such as cell titers and plate readouts will be included in this. I do also plan to implement all of our past information. Not all of our legacy data is yet stored in Benchling, and eventually this will be a catalog of a great expanse of organized past information for our legacy viruses.
Thank you, Dima. The next question, I think Ashoka, this is for you. Does Benchling currently have a roadmap for connectors, other apps to integrate more tightly with Office UI platforms like Microsoft Teams, Slack, Smartsheets, etc.? Or is the vision to build out and maintain your standard data and UI APIs and only allow those platforms to connect as a resource?
Yeah, that's a great question from Don. So right now, our plan is to build out standard data and UI APIs. However, we do have an events service that emits events via an API when things happen in Benchling. And an example of companies that have built on top of our API is people using that event system to have Slack or Microsoft Teams notifications. So let's say somebody created a request and wanted somebody to go produce a protein, that would trigger a notification inside their chat applications. So that's something they built on top of our event service.
Okay, thank you. Dima, this is a question for you. How does Sorrento deal with corrections on locked notebook entries?
Oh, okay. There's a couple different ways to handle that. First of all, entries can be retracted and edited with some communication between the reviewer and the submitting person. Additionally, I think in other cases, it can be reviewed by me. And if that person, for example, has left the company, As an org admin, we are able to open up an entry, and then we would do something like carefully note any changes we're making based on those reasons and review or re-review the entry as needed.
Perfect. Thank you. Ashoka, this one is for you. What is the best way to track if the samples were used for downstream analysis and update the samples as they were not stored in the original location anymore?
Yeah, that's a great question. So there are a few ways you can do this. You can track the movement of samples between teams using our process management tools, such as requests and workflows, where maybe you're handing a sample off to an analytical chemistry group. Other ways to track where the physical sample is located, you can check in and check out containers. So you can indicate that I'm using a sample or plan to use the sample in experimentation and put it back. You can also move it to different locations throughout Benchling, let's say to an incubator or to a different building to indicate where the sample actually is. And each of those actions that's taken on each sample is audited, so you know kind of who has touched each sample.
Perfect. Thank you. I think this will be the last question. Dima, this is for you. I'm going to combine two questions. One is, how many approximately users are there on the Benchling platform from Sorrento? And two, did you have a dedicated internal staff to help implement Benchling?
Oh, okay. Good questions. So we have close to 100 constant users right now, and that's been pretty steady from inception to current times. We do not have an internal staff to work with this. We have several org admins, myself and Dr. Allen, and a lot of these ideas were just implemented, and we were able to rather quickly implement them, and luckily they worked. But luckily, I think with a bit of organization, much of this was quick to implement. And I will also say I had many, many questions for the Benchling support team, and they were extremely helpful with that.
Perfect. Great. Thank you so much, everyone, for joining us today. We will be sharing the recording of the webinar within the next week. If you would like any additional information, please visit our website at www.benchling.com and request a demo so we can speak more about it. Also, we have our next webinar coming up next month, which is going to be high throughput molecular biology from your living room. So please feel free to register for that, and we'd love to see you there for the next one. Thank you so much.